average distance where such pattern is located.
And you can skip anything which you feel that I already covered.
Yes, yes. Hello, everybody. So I will be now discussing some practical aspects of usage of gene prediction tool using examples from prokaryotic genomes. And these tools can be used not only in prokaryotes, but also in prediction in phages, viruses, in metagenomes, in prokaryotic and eukaryotic transcriptomes, in all sequences which don't have introns. Everything which has introns will be covered in the next topic by Thomas. And there's 4 main tools I will be talking about, 4 software tools. Dr. Borodovsky just covered the theoretical background behind this tool. I will be talking mostly about the usage and interpretation of some data. We have 4 tools: GeneMark, GeneMark HMM, GeneMark S, and MetaGeneMark. These tools are supported for many years and they have multiple versions. So sometimes I will be mentioning the latest version, most frequently the latest version, but sometimes some notes will be about the older versions too. This is our website at the Georgia Institute of Technology. There's a link on the left side. If you want, you can connect to this website and run software remotely using web interface. And 3 groups where prokaryotic style gene finding is accessible is highlighted. If you don't want to run it through the website, and many times the best option is to run it locally, there is an option to download software and to use it on your local machine. And on average, every year we have thousands of downloads for software. Now, when we are talking about the software, there's always question how much users should know to efficiently use software. And what we're trying to do is to make software completely automatic, the way that user expertise is really not needed, high-level expertise not needed, and people could get high-quality gene prediction straightforward by running a gene finder. And in ideal case, this is all what user should know. He has an input, user has an input, he runs input to a gene finder and gets the output. Input is a nucleotide sequence, and output coordinates of genes detected inside this sequence. This is ideal world. Sometimes we can get better accuracy if we're adding additional information, but this is a kind of base level which I will be talking right now in the presentation. I will be switching between presentation and web interface, demonstrating how sequence runs. So for demonstration, we selected a couple of regions from the E. coli genome, and now I will stop sharing the presentation and move to the web browser so you could see how gene prediction works online. Okay, so I'm typing a web page. This is our group web page, and I'm taking a piece of sequence which comes from the, as I mentioned, genome of the E. coli, first 10,000 nucleotides, and this is a web page which gives us an access to a gene prediction tool which is called GeneMark HMM prokaryotic. We will stop in a couple of minutes later on this page more, and I will describe it in more details, but right now I just want to show very basic usage. We have the window where we can pass a sequence. I'm putting sequence aside, so you can see sequence has like nucleotide letters, and there is at the beginning label, which is ID of a sequence. We need some parameters. Here I'm using default parameters. You could see there's many species supported, some default parameters, and I want to tell what I want to see output in some format. I selected an output, and I'm running a program.
Do you want PDF or something? No.
For this moment, no. Just very simple. And prediction was done. Prediction is finished, and this is an output file. I can click on it and download. Let me make it so the way that it will be easier to see it on the screen. So we have a header on the beginning of this file, and then there are coordinates of predicted genes. The numbers which are in a column number 4 and 5 are coordinates of genes. So as you saw, the process is quite straightforward. Sequence, software, output. So now when we understand how all this system works and how quickly it works, let's go back and take a look in a little bit more details what we mean under the sequence, what kind of output formats we're supporting, and what kind of optional parameters users sometimes would prefer to set. Okay, so we're getting back to the presentation. And here we are looking on a sequence. Main input is nucleotide sequence, and it should be in a FASTA format. We call it FASTA-like because standards are changing. It's very like fast-moving field, and whatever was developed 10, 15 years ago is sometimes not suitable for today's usage. So if you will go in and look into what FASTA format is in definition, people could be sometimes surprised that FASTA supports much more than people allow it to do. So we also have some limits, and also we have some simplifications which imply less, put less errors on what user can do, because sometimes user can put a white space which really is invisible on the screen, and it will stop a program because white spaces are not allowed in FASTA format. So we allow this type of minor changes, and this is an example of really three FASTA sequences. So first sequence has ID, Contic ID, Contic_1. This will be a ID of this sequence, and you see length of the lines is different, white spaces are there. Second record is completely empty, there is no sequence at all. Third record has numbers, low cases. This is what is called FASTA-like, little bit more loose criteria on what user can supply. But if user supplies any letter which is not in this table, program will stop and fail. This is only allowed letters of nucleotide base codes. And this is something which frequently leads to the failure of the analysis when it's enough to have one character, not from this alphabet, in a 10 megabase file, and it will stop the algorithm from the generation of the output. Most frequently, we use only 4 letters and replace all the other letters by letter N, unknown. There are some rare cases when it makes sense to do more detailed analysis. If you are encountering in gene prediction some non-standard behavior, there are regions where you have to look, and one of them is input and how this input is interpreted by software. So you can check it on the documentation and see what software does. Another part of the input is genome organization. So when we're talking about gene prediction, most frequently, it's even hard to tell which kind of input is most frequent, because from point of the book, we will say, okay, we want to analyze a complete genome. But complete genome is usually available on the very late stage of the genome publication. Usually we start with a draft assembly. We're doing the analysis of the draft assembly, trying to understand how good our genome is. And then there's a later work to finish to create a complete genome. And when we have a complete genome, it can be a linear sequence. So something which we have here on the top of the sequence. There are many bacterial species, archaea species, prokaryotic species, which are circular. And when we have a circular genome, we have to split it and make it linear to put it into the database. This genome is circular in nature, but to keep sequence in database, we have to select some data point, and this data point can split a gene into 2 pieces. We have to keep this under consideration, and software should be able to predict this gene correctly. It's not 2 pieces, it's 1 gene, which just due to representation was split. And another part of the input is this, what I started from draft assemblies. Draft assemblies frequently are in a multiple contexts. We can have sometimes thousands of them. And sometimes working with a short 1,000 contexts is not very convenient. And some of the assemble algorithms are creating the scaffolds, putting context together and filling in gaps by letters N. And it's also important to understand that this is some sort of, in some sense, artificial construct. Exact length of a gap is unknown. And sometimes even orientation of the context may be wrong when we are dealing with draft assembly. So we need to know that gaps can interrupt a sequence, and they can interrupt a gene structure inside the sequence. So we can have incomplete partial genes inside the draft scaffold assemblies. And output format, there's many output formats. Standard in the field currently is one of the variations of GFF, GTF, GFF3. They are all the same names, similar names for general file feature format. And first 8 columns of this format are shown on the screen. Sequence ID, name of software, what we annotated, in this case it's protein coding gene, coordinates of a gene, and strand. These first 8 columns are practically the same between all 3 formats, GFF, GTF, GFF3. The only difference is 99% differs is in the last column 9, where additional information helps users and helps software to efficiently process these files. And because first 8 columns are the same, and we will be focusing mostly on these 8 columns, I will be using in this presentation GFF, and we will not be focusing on the differences, only on the shared part of the format itself. This format was introduced originally for eukaryotes, and for prokaryotic genomes, sometimes some deviation from the standards again could be useful. It sounds really bad each time when someone says that they deviated from a standard, because it creates a lot of complexities for software engineers and for biologists when they're analyzing, because they have to now learn multiple standards. But we do it just because otherwise it's much more complex to explain what we are predicting. So we are predicting on a sequence start codon, stop codon, there's an ORF. We are calling this a complete gene, and we should have coordinates on the left of a start codon, coordinate on the right on the stop codon, and our prediction is a prediction of canonical genes where we have all complete codons, gene consisting from the no frame shift, no introns, no interruptions, read through. So this is very, very canonical gene structure, and part of in the nature genes are always complete, like kind of most of the cases. But when we sequence genome and when we assemble genome, we can create artificial situations when genomes are incomplete due to our assembly being complete. So when assembly is incomplete, then yes, we will have some sort of a problem because complete gene cannot be shown. And in this case, in our output, we will see label, which is called, for example, partial, which will indicate that this gene is incomplete because assembly is incomplete. And let's move next and take a look now in again on example sequence from E. coli genome. And now, after we discussed in general what kind of input we expect and what kind of the output, we're getting back to the GeneMark HMM gene prediction program. So we have a sequence, we know how output looks like. There's one parameter we have to select. We need to select species-specific parameters, and we need to select a species. These parameters are specific to each species, and we will talk about how to get them a little bit later. Right now, let's assume that we have them. And frequently, some of the biologists will work with species which are already known, and models for them were already built. So this just makes some of the analysis faster. Okay, so I'm stopping here and turning again back to the website. And I'm returning back to the web page, and let's take a look on it one more time in more detail. So I can pass sequence into the sequence box, or I can select sequence from the hard drive, download a file. There are thousands of models. I'm selecting the one which matches my species, in this case, E. coli K12. Now I'm looking on types of the output, and because we discussed GFF format, I'm selecting GFF. And sometimes, not always, but when input is relatively short. And most importantly, when we want to understand what is going with a specific gene, we can take a look on a graph. Also, we can ask to create a protein sequence. We can ask also to create a nucleotide sequence files with predicted genes. And if you are running some larger task and you don't want to wait, you can put your email address and the prediction will come to your email. Usually it's so fast that in modern computers, and we're using the web server on a fast computer, you don't really have to wait much. So I'm starting the prediction and it's done. I selected 4 types of output. And first output is GFF format, coordinates. We already looked on it, and 2 critical columns are this column with coordinates, and this is a strand. Okay, let's take a look on a file with protein sequences. Yes, each gene predicted will end up in a protein. We have a gene ID and a full sequence starting from start codon, but stop codon is excluded. And on the top of each file, you will see some information about length of the protein, location of protein on DNA, so you can track back and use this information if you are interested in some relative location of proteins. Similar information is available in nucleotide level if you want to do some multiple sequence alignments or some blood search. You have a gene, you can submit it from here to the next step. And now let's take a look on a graph. And I want to emphasize that this graph comes from different tool. It comes from the GeneMark, not GeneMark HMM. So what we do, we join 2 different algorithms together on the website. So for end user, there will be much more informative way to look on the data. Yes, so predictions by GeneMark HMM are these thick black lines. In this case, it's a long gene on direct strand, and we have on the first 3 columns, panels show a direct strand. This continuous line shows open reading frame. It shows that there is no in-frame stop codons when we are reading a sequence. This is a start codon, we are following start codon, and there is no stop, and we are reaching on the next page. Here is end of this gene, and then next the gene starts. So this thick black line is prediction of the HMM. This thin black line is prediction of GeneMark algorithm. And this number, this black line, values vary between 0 and 1. And when we see that value is 1, our interpretation is quite simple, that probability of a fragment centered around this point to be coding is almost 100%. So when you look on this graph and you see that this line goes almost always along the 1, you are getting some sort of statistical confirmation, which is really like visual confirmation that this gene prediction is very reliable and has probability 1 almost across full length. Sometimes probabilities can drop down. For example, if I'm looking on this gene, you see that this gene has a length roughly around 300 nucleotide, but high coding potential is only across the half of a length of a gene. It still is a gene, it's very well known, annotated, predicted, it's correct prediction, but still statistically, when you look on it, you could say that frequency of letters used in this gene to encode this gene is a little bit away from optimal. So when you look on the probability graph, it doesn't go the way how it goes, for example, for a gene which is left to it. Yes, so you see it's just black line goes just straight along the full length of a gene. So this visual interpretation is very useful when you are working with a specific gene and when it's important to understand why this gene is missing or why it's predicted and what is the relationship between the genes which are near each other, do they look similar? But as you see, there's many pages. So if you are working with a genome which has 4 or largest prokaryotic genomes will be having like 12 megabytes, there's really no way for a small team to go manually through all of the pages. Okay, so I'm switching back to the presentation. Okay, I'm switching back to presentation, and we already know how this web page looks like, and this what we were discussing, that we have input in a FASTA sequence. We selected a model for this specific program GeneMark HMM and created output. Similar way we can do the input output with a program which is a GeneMark. We will not go into all the details, but by looking on the web page, you see that it looks almost identical. There are some parameters on the bottom which we usually don't recommend to change. Just keep them default. Only in rare cases, changing of them may improve visualization of the data. And this algorithm follows exactly the same approach of getting a sequence, selecting a model, and predicting a gene. It's very convenient to use the website, but there are a couple of limitations. One is software is capable of doing much more. We have more support for more options, and when you are working with single conjugate, it's okay to submit it through the website, but if you want to analyze sometimes hundreds of genomes, sometimes thousands, in many projects, sometimes we run 12, 15,000 genomes in a day, then you have to download the software and install it locally. And there is another limitation related to a graph, that because graph is usually used for a single genome, single sequence, single gene analysis, graphs are supported only for single conjugates. So if you have multiple conjugates, you have to split them to get graphs. And when we move to a download, in a usual Linux environment, we are supporting not only Linux, but also Mac OS, but Linux is default. Pretty much any flavor of Linux should be okay. And if you look on the left part of this panel, you will see a name of the program, which is like GeneMark HMM prokaryotic version 2. And we need to provide an option to give a path where parameters are, input sequence file, where to put output, and format. So this top shows very straightforward way of specifying exactly the same thing which you did, which we just did on a website. There are some additional files. For example, I can say what kind of a genome I have. Is the genome linear, circular, or fragmented? Sometimes when there's too many conjugates and I'm interested in some specific conjugate, I can tell, give me this conjugate, or on opposite, ignore this conjugate. And if genome is really large, I want to see how that program is working, so I can ask to show the progress bar. So you see just some more convenient thing to run on command line. Full list of options is much larger. You can run a program with option --help, and it will print on a screen all possible options for this software. Now, when we move next to the metagenomes, this jump between single genome and metagenome, in case of a single genome, I mentioned that we have to take a pre-trained parameter file for E. coli. And I will be talking about how to train it later. But to run the software, we need a pre-trained parameter file. The same story with metagenomes. We need pre-trained parameter file to predict metagenomes. Procedure which is used to build this pre-trained parameter file is very different, but this is done once. There is a file, and for each new metagenome, you are just using this file. You are not rebuilding the parameters. And metagenomes are usually large. Small metagenome is somewhere around 50 meg in my experience. And large metagenome is around gig or 2. So if you will try to submit a 1 gigabase sequence to our website, it will handle it, but you will spend probably half an hour just uploading and then downloading a sequence. Doesn't work. Please just download the software, install it locally, and run it locally. It runs really very fast and gives you much more convenient way to use the software. Now, what is metagenomes? Before we had the sequences from one species. Now we have a sequence for multiple species in the same file. And to simulate metagenome, I selected a sequence from the E. coli and from Bisaptorus. And I will now copy those two sequences and submit a prediction, again, switching to a website. Okay. GeneMark. So I need a sequence. Usually with GeneMark sequence will be quite long. So copy paste method is usually not the best one. Best is to use sequence upload. Okay, but right now I'm just using a short example. I'm pasting sequence. So now I have sequence in this box, and because this is metagenome, I don't have any more an option to select a parameter file. It's already there. Yes, and I'm asking to create output in GFF format. I will ask also to create a protein sequence, and you see there is no graphs. And the reason that there is no graphs, because usually in metagenomes we have multiple contexts. So it's not something, as I mentioned, useful in this case. So I'm starting a program and we are getting a prediction. With usual short context with prokaryotic genomes, you don't have to worry much about using our web page. When you are running a metagenome, story is a little bit different. Sometimes people will submit very large sequences. When you submit a sequence, you have to record for yourself this job ID. If there's any problem, we can't help you without this job ID. Then I can go. If I have a job ID, I can go down and see what went wrong. Without it, all information is deleted. We are not following any user. So I don't have a way to tell what did you run on our website. So if you want to ask a question, please send us job ID. Let's take a look on output. And again, GFF file looks the same way. What we want here to emphasize is this. First sequence, I just highlighted it, was from the Bacillus genome. And model, which was selected to predict it, genes, has GC composition 42. So metagenome analysis software took a sequence, determined what kind of a model should be used for this specific context and made a prediction. And for next context, which is from E. coli, GeneMark used a different model, which matches a GC of 52%. And it made a different prediction. Yes, and I know if you probably don't see these numbers, but I know these numbers. It's the first gene in E. coli. It's identical. So correct prediction for all the genes. For each context, the corresponding GC model were selected. And if I will look into amino acid sequences, nothing really changes for end user. He sees pretty much the same file with the proteins. The only thing you can like kind of track the idea of a sequence and say, okay, this is this context, this is that context, but all the predictions we can just submit from here to BLAST, and they come up back with a high quality annotation. Okay, what changes if I have like 100 megabase or 1 gigabase genome? Really nothing. It's as simple as what I just did with the 2 short sequences. The only thing is you have to wait somewhere around like 15 minutes if you're running it on a website. You will wait some minute or 2 if you're running it on a local computer. Okay, so I'm stopping web page and getting back to the presentation. Okay. Okay, so we are back to the presentation, and we are looking again on this general concept: input, model, output. With GeneMark, you even don't see model files. Yes, so it's just somewhere in the background, all done, so you will see an output and prediction. But where we are getting parameters from? The species parameters, which we just selected on the previous pages. There is an algorithm which is called GeneMark S, and then newer version GeneMark S2, which does solve both tasks. It takes a genome, estimates parameters, and makes a prediction. And majority of the, I would say almost all models which we have on our website were built by GeneMark S2. So we build them, we put them on the website, so end user can just select a species. You have to just make sure that when you are running GeneMark S2, that input sequence is coming from the same species. It's not a metagenome annotation, it's an annotation of isolate. And more sequence, better quality assembly always leads to a better annotation. It doesn't mean that you can't run a draft sequence. Annotation on a draft sequence is frequently quite close, but general kind of approach is with self-training, with parameter estimation tools. More sequence is better, more continuous sequence is better. If code fails, usually it fails because there is a assembly quality is low. There's not enough sequence. Okay, so we are moving next, and I selected an example to show how GeneMark S2 works. In this case, example is complete genome of Bacillus subtilis. And I did run it on a local machine using this command. And you could see what this command really is doing. I'm telling program to use sequence, and this is a name of the Bacillus subtilis assembly. I'm telling that it's bacteria. It's not necessary to tell it it's bacteria or archaea, you can set this variable to auto, telling that it's bacteria or archaea. Sometimes speed up the process, really speed up the process a little bit, but usually users know what they are dealing with. If you don't know, you can set it auto. I'm telling what kind of output format I want and output and file name. As you see, again, there's really no expertise required. Almost all options are very straightforward. And you can go in and run this on a web page. I don't know if it shows on your screen, or do you see still, what do you see on the screen? Do you see the PowerPoint presentation or website?
Still the presentation.
PowerPoint.
Okay. Okay, sorry. Okay, so runtime is 4 minutes, so it doesn't worth of spending time, but I just want to still show how this page looks like. Okay, so hopefully you see now a website. So again, the same style. I can choose a file, and it may be okay. I don't. I think this one is Bacillus subtilis. Yes, I'm selecting a genome from the command from the option, and I'm leaving, like I can ask, okay, give me in a GTF format, prokaryote, genetic code 11, and I usually ask also protein sequence and start. Now you see that it is working, and there is an estimate of time, how much time at this moment it estimated will take to train. So I'm stopping sharing because it will take a couple of minutes, and I'm going back to presentation. And back to PowerPoint. So what we will get will look something like this, what is right now on the screen. Again, the same GFF format. Header on the top has information about the version of a program. If there's some issue, some problem, you see that something is not working the way how it is expected, this header is important for us. Please send information to us about it, saying that, okay, program failed with this command line, and this is in a header. And then for each content in the assembly, there is again, in one or another form of GFF format, we have a coordinates of genes, strand, and what program did, it trained from sequence alone all the parameters and made the prediction. What interesting we can tell from this output? Pretty much it's a prediction. It's what goes into the annotation. If you are looking to specific gene, you are starting, it's your starting point. But sometimes we can ask another question. We can say, okay, what training showed us interesting about this genome? And we can go in and look into inside the model file. So model file is created for each training. And what it tells us that this genome belongs to a group A. Information about which group, what it means, is in a GeneMark S2 paper. And group A corresponds to a group of genomes which have canonical Shai-Dang-Gano RBS translation initiation mechanism. And what we have on the screen right now is a logo of motif which was found by GeneMark S2 training. And this is this AGG. It matches perfectly the mechanism of Shai-Dang-Gano. We didn't give much of information. We didn't really told the software that we want to predict group A genome. It's a software self-trained told us that this is a group A genome and that this genome is using the Shai-Dang-Gano sequence. And most frequently, I would say the key file to look on success of the training is the graph on the left. This graph shows a distance between the Scheidel-Garner signal and start codon. And you see that this graph has a max around 7 nucleotide, has a shape of the bell shape. So there is a preferred location. And when we are moving closer to start codon or further from start codon, frequency of the events of Scheidel-Garner dropping. So again, the information about the shape was not given to a software. It learned on its own. And because it learned on its own, it gives us some assurance that, okay, it matches what we expected biologically. We expect that signal should be somewhere in the front of the 7-8 nucleotides with some variability, a little bit closer, a little bit further. We can't, without any additional information, verify that all our prediction is correct. But indirectly, by looking on these graphs, you can make have some peace of mind that what your algorithm found corresponds to expected biological behavior. Okay, let's take a look on the another option. This time it's Mycobacterium tuberculosis. It's a different genome and identical command line. Yeah, so name of a sequence, genome type bacteria, format, and we're running. Program again takes somewhere about 4 minutes of time. If we will look inside the model file, it tells us this. It tells us that this genome belongs to group C. Group C, if we again will look into the GeneMark S2 paper, are bacteria with a significant number of genes which use leaderless translation initiation mechanism for protein translation. And we again can go inside the model file and using model file build two graphs. Some genes in tuberculosis are initiated, translation is initiated using canonical Scheidel-Garner RBS signal. And we see this signal on the top. Again, AGG, AG, so it is matching expected, matching 16S RNA tail, it matches biology. I want to emphasize again, we didn't told that this species is using this mechanism. It was found by algorithm itself. And then when you look on the length distribution, it has a peak around 6. Shape again is bell, quite close what we will expect. And second part of this panel shows a promoter. So 25% genes in bacteria, Mycobacterium tuberculosis, are first genes in operons, which are initiated, translation is initiated using leaderless mechanism. So there is no signal on a sequence for 16S RNA to bind. Sequence starts with a start codon. And in this case, signal which is associated with a start is a promoter. And we see classical canonical promoter signal with a lot of T's and A's for this type of a bacteria. And when you look on localization, it's perfectly localized in a fixed position. Almost all the signals are in a all the promoters are and position from 6 to 8. And as I mentioned, transcript should start from ATG, from start codon, for this mechanism to work. So there's significantly less variability here. And signal is much, much stronger if you will compare like top graph and bottom graph. Frequency is 0.4, yes, here. So this is interesting biological observation, which is derived by unsupervised training. And this information on the screen is used to improve gene start prediction and overall gene prediction for users. Okay, we can take a look on a similar signal from another species. I will not stop here for long. just to show that it's an archaea, and in archaea we may have also RBS and quite a different kind of the promoter, because it's archaea, it's not bacteria anymore, it's located really far from the start, around 25 nucleotides. But again, what I want to emphasize here is that by just running a software and looking into the model file, we can make a conclusion about how successful training is and if some interesting biological event was observed. So question which sometimes I'm getting, and we are all getting, is like how we can tell that algorithm is performing very well. The only way to tell is to have high quality test sets, and they are very difficult to get. There are very few genomes in prokaryotic world which have high quality annotation for all the genes. Practically E. coli is the only genome probably where we have a lot of high quality annotation, and only in direct way we can look on a genome and say that software did found a correct answer. Mostly we are basing our conclusion on a match between the expected and known biological nature and what algorithm predicted. Okay, so I have probably another like 2-3 minutes left, and now we are moving to a next step. I mentioned that at very beginning that the only thing which we want to use during the gene prediction is sequence, and no expertise from end user. Yeah, so sequence only and no expertise. Please run a program, get prediction, and everything is done by machine learning behind the web interface. But sometimes we can do a little bit better. We can improve the prediction by taking additional information into the software, and by this improve the gene prediction. So GeneMark S2 is used as a component in NCBI's protein genome annotation pipeline. All the genomes which go to NCBI into the RepSeq project are automatically annotated by this PGAP pipeline. Moreover, if you want to submit a genome to NCBI and you don't want to spend time on the annotation, there's an option on a submission website in a gene bank that you are asking NCBI. They do it for free. They do it for you to annotate your genome, and they will run this pipeline. And here you see in the corner, there is a GeneMark S. It has a plus here. Plus here stands for this additional input. So additional input comes from the information that some conserved proteins are very easy to find and very easy to find by using like similarity research tools like BLAST and alignment tools like ProSpline. And these tools can create additional hints for GeneMark S2, so it will create a prediction which matches current knowledge about this genome. And this is this input, additional input, which we are getting into GeneMark S2. And then our prediction is merged together with prediction from the similarity search, from homology-based prediction. It is merged with RNA-seq, with RNA gene annotation. So there's a lot of components going in, and you are getting the final prediction. So full process is described at NCBI. You can download, if you want, a code for PGAP and run it locally. This is a resource and time-consuming pipeline. It's very high quality. It does a lot of not just structural gene annotation, but also functional gene annotation. It will give you a functional labels, but because it does functional gene annotation, it runs a lot of additional analysis. So you could expect that every genome of size something like 4 meg will run 5-6 hours, while GeneMark S2 will give you prediction in a couple of minutes. Installation instructions are quite reasonable, but you have to follow them strictly. If you don't, big pipeline will not work. Yes, it's not, but they are very responsive. If you have a question, you can send the question to the on a GitHub, and answer will be given you quite fast if something doesn't work. And okay, so 1 more minute and I will finish. At very beginning, I mentioned that this software works not only for prokaryotic genomes, but also from viruses, phages. This is what was done was GeneMark S2 was used for the prediction in T4 phage, and size of T4 phage is only 168K, and this is the RBS signal derived on the left, and localization of the signal, spatial distribution between start and RBS. It just, again, minor visualizations that you run it on a shorter sequence of phage, and it gives you correct answer, expected answer. So summary of this kind of components, what I want to emphasize at the end, that software is automatic. You still need to provide some metadata. You have to tell what kind of genome came out from the sequencing machine. We can't guess it on our own. So you have to provide circular, linear, or incomplete. Yes, we are doing some deviations from a standard because GFF standard doesn't have anything inside about the incomplete or complete genes and how they should be annotated. So please check the formats before you use our software. Graphical output is very useful if you are working with a specific gene, specific group of genes. Please use, I'm always using myself. It gives me very intuitive understanding what is going on. But please don't use it on a large genomes. You will just run a website for nothing, because it's really impossible to go through all the graphs. I want to emphasize it's not always like that. There was a case of first genome annotation of the Bacillus subtilis genome in France. And I know that they printed all the graphs of GeneMark, and they had a big room where whole genome was on a wall in the form of the GeneMark graphs, so they can stand and look on a specific fragment by eye. It was like a long time ago, so when computers were not able yet to do this type of visualization, but they really had access to this type of information. So you can do everything locally, you can do everything through the website. Preferred option is, I'm recommending everybody to use local installation. If you are doing more than 2-3 genomes, I am frequently using web page because it's convenient. You are all welcome to use it. There is no any limitation. And before we move to the eukaryotic part, I want to emphasize some difference in the community. So when we talk about the gene prediction in prokaryotes, requirements are quite different from what is in eukaryotic world. For example, if there are repetitive sequence, in eukaryotes, it's masked. In prokaryotes, no. In prokaryotes, when there is transposable element, people expect that genes prediction software should predict correctly protein encoded by transposable element. This is not the case in eukaryotes. No one is really looking into annotation by gene founder of transposable elements in eukaryotes. When we deal with pseudogenes, and when we are talking about how well pseudogenes are predicted by gene finder, this information is completely removed in eukaryotes, because really the people are not discussing this. In case of prokaryotes, there is this pressure. It's important to, from software, to be able to match pseudogene as close as possible. And similar thing about the phages, which can integrate and create additional complexity for genes binder. So even if, like eukaryotic world, we have much larger genome, more complexity in a prediction due to introns, there is a completely different requirement set in prokaryotes. So these additional components, no masking, transposable elements, pseudogenes, phages, they create complexity which is not in eukaryotes. So it's a very, you can directly compare two words. And one more component is minimum gene length. It's not on the screen. Minimum gene length in prokaryotic world is somewhere around 90 nucleotides standard right now. Few algorithms are capable of predicting shorter genes correctly without much of false positives. We are keeping it at the level of 90. If you are comparing two gene finders, you have to pay close attention what the minimum gene length is. It's really easy to change the balance by changing the balance of minimum gene length, and it can bias the comparison. Okay, if you want to use website, there is a web page, and if you want to contact, there is a web page. If you have any questions, any problems, please contact us. We will try to do everything what we can, so software will work and work correctly. Thank you very much, and if you have any questions, please.
Hi, Dr. Alex. Is it right?
Yes, Alex is correct.
Okay. Well, I wanted to know, how does GeneMark handle a sequence with many repetitive regions?
I'm sorry, there was an interaction of two people talking, I think, and could you please repeat the question?
Yeah, sure. I said, how does GeneMark handle a sequence with many repetitive regions?
Mm-hmm. So GeneMark will, in case of prokaryotic GeneMark, it will predict a gene in each repetitive sequence fragment. And this happens, for example, in even in E. coli genome, the insertion sequences, elements, kind of a transposons. And we will have 8 identical predictions. Yes, if there's an 8 transposon, we will do the 8 identical predictions, and you will get like 8 additional genes. This is already on the side of an end user. So when you have a prediction, you have to compare genes in between and check that, oh, these 8 are really the same gene. It's not 8 different genes. This is a component. In terms of a training, it's a little bit not as straightforward because it comes down to a length of a repetitive sequence and length of the genes predicted in repetitive sequence. Short genes are not used in training, and these short genes are frequently predicted in repetitive sequences. So usually repetitive sequence is not influencing parameter estimation. So you may be safe on that side, that you don't have to mask it. But again, if you want to mask, you have this option. You can put all the letters in uppercase, and you can soft mask and put repeats into lowercase and tell program, please use soft masking. And then in regions with masked repetitive sequence, there will be no prediction.
Okay, thank you.
Okay, any other questions?
Me again.
Yeah, sure.
I saw some values to the right of the termination codon position in the output. What do they mean?
I'm sorry, there's a problem, something with my connection right now. Could you please repeat again? I kind of heard only the beginning of a question.
There are some values to the right of the termination code on position in the output. What do they mean, those values?
Okay. So let me see, depending on the, thank you for bringing this up, because this is something I wanted to talk about, and okay, so question is about the additional columns which we see on the screen. So this is coordinates X, Y of a gene, left, right position. And this number is after, it's a logit score of a gene. This logit score can be used as a some measure of a reliability of prediction. If you say that number is large, then this gene is probability of this gene to be correct is really, really high. So you could see that the first gene has really small number, close to like 1, 0. It may look like a false positive. And after that, genes have a higher number. Yes, so, and this number is proportional to length of a gene. Longer gene, it will be a higher logit score. Now, if I'm looking to the right side of the column, this is column 9. Format of the first 8 columns is strictly controlled. You can't really change meaning here. Last column, there's a ways to change meaning and add additional information. What we have here is a gene type, and gene type sometimes can be interpreted. Dr. Borodovsky showed a slide with information about native and atypical genes, but in this case, like following his slide and explanation, native genes, it's genes which are using the codon usage frequency, which is shared for a full species. It looks like this gene is from this native genome. And there's a gene which is atypical here. You see it's, sorry, short one, relatively. And you can pay attention that it has really different GC composition. Majority of genes is 40 something, and this gene has a 37. So it's very possible that this gene came from some virus or a phage. It's a lateral transfer. It's not a native gene. It moved to this genome from another place. It's one of the explanation, and there's a reason why we think this, because it has very different GC composition. Yes, 37, and it has very different label, atypical. So in a column number 9, we can give much more additional information. We can give sometimes there are some command line options you can use and get very detailed information, how good start prediction is, what is the score of the RBS, what is the score of promoter. So if you want to zoom in and look on a gene level, there is much more data. But basic thing which we are looking into is coordinates, and I would say that this logit score is very informative when you are making a decision if you want to work with this gene or not. Hope that this gave an answer or question.
Yeah, absolutely.